The story of SpongeBob and the point mutation that changed his life forever. Meet SpongeBob, a square who's yellow and porous. He lives under the sea in his pineapple house with all of his friends. He also has a pet snail, Gary. Now, let's talk about his DNA, RNA, and his genes.
The first step in this huge process is called transcription. Transcription is the process of making an RNA copy from a DNA template.
This is a DNA strand. It has a promoter on it. The promoter will tell where the polymerase should bond. Then, just like helicase, the polymerase will unzip the DNA strand. So, this process can begin. The antisense strand is a strand used as a template from the DNA for the polymerase to make a complementary RNA strand off of. The codons are based off of each other, so the RNA can be formed. Here, the polymerase is slowly starting to make the complementary RNA strand based off of that antisense strand that is again used as a template. Once the polymerase is done making the strand, it simply falls off. The process of transcription is over. Now let's talk about the next step, mRNA editing. mRNA editing is the process in which an M or messenger RNA strand is changed and had some things added to it in order to make it mature so it can go on to the next step to making a protein. Here is an mRNA strand with introns and exons. Now, the first step is where the introns all get cut out and the exons get spliced together. Then a cap and poly-A tail is added. Now that that's over, let's go to the next step. The next step is labeled translation. Translation is a process in which a ribosome reads the now mature mRNA and makes a protein or a polypeptide chain out of it. There are two parts in this process, mRNA and ribosome. The ribosome has two different sites, a P site and an A site. The P site is a polypeptide site, and the A site is an amino acid site. These are where the tRNA goes into. Now, after these two things run into each other, the mRNA goes inside of the ribosome. Here is tRNA. As you can see, it has an amino acid on top and anticodons on the bottom. Here, the tRNA goes into the start codon sequence on the mRNA. This tells the tRNA where to go into. Now, since it's in the A site, it can start the process. Now, it scoots over and another tRNA goes into it. Now, the tRNA is broken off and the little amino acid creates a chain. This goes on and on between all of the different tRNAs that go in once it reaches the stop codon. Now we have a polypeptide chain. A mutation has occurred. A mutation is any kind of change to the base sequence of either DNA or RNA. Here, in SpongeBob's RNA, a point mutation occurred, which affects a particular gene only. Here, substitution is happening. Substitution is when one base replaces another. This doesn't seem like it could really do much damage, but it can really take a toll on an organism, like SpongeBob. Let's see what happened to him. Here's the new SpongeBob. He's not porous and square anymore. He's a round yellow sponge with no holes. His friends don't recognize him anymore because he's normal, all because of this one point mutation that changed his life forever.